Hello students. Today we are going to start the next chapter, that is the cell. So many living organisms exist in this earth. The body of all living organisms are made up of cells. So a cell can be defined as the smallest structural and functional unit of life. If you think of a house, what are the building blocks of the house? Obviously bricks. Like that, cells are the building blocks of all living organisms. Living organisms can be classified into two types. One is unicellular organism and another one is multicellular organism. What is unicellular organism? Organisms that only have a single cell. You can see here all the examples of unicellular organisms like amoeba, bacteria, paramecium. Now what is multicellular organism? Organism consisting of many cells or multiple cells. Most of the plants and animals are of this type. Next, we will proceed to the discovery of cell. Cells were discovered in 1665 by English scientist Robert Hooke. He made a simple microscope to observe different objects. Once he had taken cork cells to see the structure. Actually, cork is the plant cell extracted from bark of plant. While he kept a thin slice of cork under his microscope, he noticed some box-like structures looked like honeycomb. He named these boxes as cells, which means little rooms in Latin. This is the microscopic view of the cork cells. Actually, cork cells are dead cells. So he observed only the outer cell wall. You can see here the cells are mainly hollow or empty. Only the outer cell walls are visible. Almost at the same time, another scientist, Anton van Leeuwenhoek, made more powerful microscope than Hooke's microscope. Using this microscope, he examined so many things like water drops scraping from his teeth, and by that way, he had first observed single-celled organism, that is bacteria. So, the term cell was first coined by the scientist Robert Hooke, and the single-celled organism bacteria was first observed by the scientist Anton van Leeuwenhoek. Next topic is cell theory. By the early 1800s, scientists had observed many cells of many different organisms. These observations led two German scientists named Theodor Schwann and Matthias Jakob Sladen to propose that cells are the basic building blocks of all living things. Actually, Sladen, a German botanist who viewed plant parts under a microscope and discovered that plant pods are made up of cells. He is considered to be the co-founder of cell theory together with Swan, with whom he consulted. Theodor Swan, the second scientist, who viewed animal parts under a microscope and discovered that animals were also made up of cells. He extended Sladen's cell theory in plants to animals, stating that all living things are composed of cells. One more scientist, Rudolf Virchow, stated that every cell originates from another existing cell. Virchow demonstrated that the first evidence of cell division, and he proposed that living cells arise only from other living cells. The ideas of all three scientists, Swan, Sladen, Virchow, led to cell theory. Cell theory includes three principles. First is, all living things are made up of one or more cells. Second is, the cell is the smallest structural and functional unit of life. And third is, all new cells come from pre-existing cells by the process cell division. In early 1900s, a new type of microscope was invented called Electron microscope. It actually used a beam of electron instead of light to observe extremely small objects. With an electron microscope, scientists could finally see the tiny structures inside the cells. By that way, they can understand the structure and functions of cell organelles. The first electron microscope was designed by Max Knoll and Ernst Ruska. According to the magnification power, electron microscope is much more powerful than light microscope. This is the picture of modern electron microscope, and here you can see the comparison of magnification power of light and electron microscope. This is the image observed under light microscope, and this is the image of electron microscope. So by seeing only you can understand the difference of magnification power. Electron microscope is much more powerful than the light microscope. Now next we'll discuss about the size and shape of the cells. Cells are mainly microscopic. Their size is measured in microns or micrometer. 1 micrometer is equals to 0.001 millimeter. Average cell size is 3 micrometer to 30 micrometer. In this chart, you can see the different cell size. The smallest cell is the bacteria cell. Size ranges from 0.2 to 0.5 micrometer in diameter. In our body, smallest cell is RBC or red blood cell, which measures 7 micron. Largest cell is ostrich egg. Here you can see it is the largest egg, which you can see by your naked eye. You don't have to use any of the microscope to see that ostrich egg. Size of the ostrich egg is 170 into 135 millimeter. In our body, longest cell is the nerve cell. Cells vary in shape also. In this chart you can see so many types of cells having different shapes. Like WBC, that is irregular in shape, which can change their shapes according to their function. Here you can see WBC. Whereas the nerve cell assume a definite shape. This one is the nerve cell. Cells may be long like nerve cell or circular like fat cells. This is the fat cell, this is the long nerve cell. They may be oval, rectangular, spindle shaped, cylindrical, so many types of shapes are there. Actually, shape and size of the cells are related to the functions they perform. Cells are of two types, prokaryote and eukaryote. In this diagram you can see this one is the prokaryotic cell, this one is the eukaryotic cell. Prokaryotic cells are the primitive cells. Here the nucleus is not well developed and it lacks membrane bound organelles. Example is bacteria cell. This is the structure of the bacterial cell. Here you can see the cell membrane, cell wall, and one more layer is present out of the cell wall, that is capsule. And here you can see no membrane bound nucleus is present. In place of it, that is present nucleoid. Actually, nucleoid is the primitive nucleus which is not bound by nuclear membrane. Now next is eukaryotic cell. Here you can see that the nucleus is well developed and so many membrane bound organelles are present inside the cells. Here is the difference in between the prokaryotic and eukaryotic cells. In prokaryotes, no membrane bound nucleus is present, and in eukaryotes, membrane bound nucleus is present. Prokaryotes cell walls are made up of peptidoglycan, and eukaryotes, if cell wall is present, it is made up of cellulose. In prokaryotes, no membrane bound organelles are present. In eukaryotes, membrane bound organelles are present. Like that, cell size varies also. In prokaryote, cell size ranges from 0.5 micrometer to 100 micrometer, and in eukaryote, cell size ranges from 10 to 150 micrometer. So by that, today we are ending our session. Detailed cell structure we will discuss in our next session. Till then, bye and have a nice day.